Hello there, and welcome back to Peter's Primetime Practice, where we are going to look into the mitochondrial DNA analysis in forensics. What is mitochondrial DNA? Mitochondrial DNA, also known as mtDNA, is a small circular DNA molecule located in the mitochondria. It is approximately 16,569 base pairs long and consists of 37 genes and a control region, also called a non-coding region, since it does not code for any gene products. Mitochondrial DNA analysis is the perfect fit for the field of forensic science, especially when creating DNA profiles from types of evidence that might not be appropriate for other DNA analysis techniques such as restriction fragment length polymorphism or short tandem repeat analysis. There are 6 simple steps in the process of mtDNA analysis. Step 1. Primary visual analysis. A summary of what occurs in this step is that basically scientists examine all the samples such as hairs or any type of sample needed for analysis and run them through tests making sure that they are of human origin. This is important because mtDNA analysis does not run if the sample isn't of human origin. Step 2. Sample preparation. In this step, all that is done is all the samples that are gathered are just cleaned and made sure to be safe before beginning mtDNA extraction. Step 3. DNA extraction. The DNA that is extracted out of the sample, it is then exposed to a mixture of organic chemicals that separate the DNA from other molecules such as proteins. Step 4. Amplification by the polymerase chain reaction. First off, PCR is a procedure that makes many copies of small amounts of DNA. Therefore, in the first step of the PCR process, heat is used to separate the 2 strands of the DNA. A new DNA strand is then made from each template using an enzyme that copies the existing DNA molecule. This copying process is repeated a number of times, and during each cycle, the amount of DNA in the reaction tube is basically doubled. At the end of this process, millions of copies of the original targeted DNA in the extraction are present. Step 5. Post-amplification, purification, and quantification. The DNA created from the PCR is then purified using filtration devices that remove excess regions used in the PCR from the sample. Using capillary electrophoresis, the quantification step is performed. If by any chance the PCR failed to yield the product, it can then be extracted and amplified again. But if it is successful, then the products are prepared for DNA sequencing. Step 6. Sequencing. In the final step, Sager's method is used for mtDNA cycle sequencing, which is quite similar to PCR but slightly different chemicals are used. After this method is completed, gel electrophoresis is used to determine the sequence of the DNA. Once that is complete, the process of mitochondrial DNA analysis in forensics is done, and now time for the scientists to wrap up their thoughts and send their conclusions to the cops, where they use that information to catch the bad guys.
Yeah. Hey.